Good morning, Cougars. I want to spend a little bit of time here at the beginning of the school year talking about COVID-19, which is, of course, affecting everything that we do this year. It's important in a life science class, biology or zoology or AP biology, to talk about that because this really applies to what we're studying. This is all about um, an organism that infects living things, and we're living things, and that's why we're having to work so hard to try to avoid being infected by this. So here's a picture of what the virus that causes COVID-19 sort of looks like. You can see it's got some spiky things sticking out of it. Let me back up a second. There we go. And we'll talk about that in just a minute. So viruses are a part of the living world. Although it can be argued that they're not really alive, viruses cannot carry out most life processes by themselves, but they hijack the cells of their living hosts to allow themselves to be reproduced. Because of this, they're made up of the same kinds of materials that living things are made of. So they are made of two basic parts: an outer coat made of lipids and proteins, and then some kind of genetic material, either DNA or RNA, on the inside. And it's the genetic material on the inside that will force the virus's host to use the cell's machinery to make new viruses. So in this diagram here, you can see we've got this outer layer here. This is what's called a lipid bilayer. Your cells have a lipid bilayer on them also. And then there are these proteins sticking out of them. And that was the spiky things that we saw on that first diagram. In fact, they're called spike proteins. Okay. So the spike proteins in the lipid bilayer of a SARS-CoV-2 virus. By the way, SARS-CoV-2 is the name of the virus that causes COVID-19. It's related to another virus that caused a disease about 20 years ago called SARS. That was SARS-CoV-1. So it's related. The way that it infects people is a little bit different. Okay. The spike protein attaches to a receptor on the protein in the host cell called an ACE2 receptor. The viral RNA then is released into the cell. Okay. The viral RNA then is used by the host cell's endoplasmic reticulum to make new viruses. So this is just the same process that the cell goes through with its own RNA, but in this case, it's taken the viral RNA and it's using it, and it's creating viruses instead of the parts that it would use to carry out its own cell functions or to copy itself as a cell. Once those viruses have been made, they escape through the plasma membrane of the cell.